Good morning, everyone. Today we are going to continue with the revision on genetics. The main topics we are going to deal is the deviated results of inheritance pattern of typical Mendelian ratios. So, the first topic coming under that is incomplete dominance. So, after Mendel, when many scientists when they conducted the experiments, they got deviated results from the typical 3:1 ratio. So one such example is incomplete dominance. So what is incomplete dominance? It is the failure of dominant allele to mask the expression of recessive allele completely in heterozygous condition and does not resemble any of the parent the F1 generation. That means in F1 generation, a new phenotype can be observed, which does not show any resemblance to either of the dominant parent or of the recessive parent. And as a result, when they conducted F2, they got a deviated result, that is 1:2:1. Typical example is Snapdragon antirrhinum or 4 o'clock plant Mirabilis jalopa. There were two contrasting forms, and when they crossed these two contrasting forms, that is flower color, red flowered plant when they crossed with white flowered plant, in F1 as per Mendel they have to get red, but instead of that they got a mixed up character. That means a blended expression was observed. The F1 plants they produce the pink flowered plants, and this pink is not at all resembling red parent as well as the white. So a blended expression was able to observe in the F1, and on selfing this F1 pink with pink and F2 they got 1:2:1, that is 1 red, 2 pink and again 1 white. So from this they concluded that there are certain deviated results in which in that cross the dominant recessive relationship will be lost, and even though both the alleles will not blend, when these two contrasting alleles are coming together, none of them will resemble their parent. Instead of that, they reflect a blended expression in the F1. And that F1 you can see in this figure, red into white, that is capital R, and here it is small r. That can represent, or that the gametic combination we can represent with white capital R, then white can be represented with small r. And here the F1 when they are getting they were showing pink color, and the gametic representation also we can represent in this way in circle. And when we draw this Punnett square, the phenotypic ratio appear in the form of 1:2:1, as well as the genotypic ratio appear is in the same pattern, that is 1:2:1. That means one is red, that is purely homozygous. The heterozygous, instead of showing the dominant character, they showed the intermediate trait, that is pink color. Then the last one, it is recessive color, that is represented in the form of white. So this is a typical example for incomplete dominance. Now we'll move on to codominance. That is also another deviated result of Mendelian typical ratio. Here also the ratio is 1:2:1. But the difference is, in this condition, both the alleles, when they come together, here also they will lose their dominant recessive relationship. But in the heterozygous condition, both the alleles express simultaneously. That means they resemble both the parents. Here, the typical example we can give the blood group, that is AB blood group, and even carrier of sickle cell anemia, coat color in cattle. The ratio in F2 ratio here also it will be 1:2:1. So here when we move around the example, typical example, codominance, it is AB blood group. You should not say ABO blood group. In ABO blood group, AB blood group alone only following the codominant relationship. So as per it is based on a type of sugar which is present on the plasma membrane of RBC. So here I have represented the RBC. Then on this RBC there can be two different types of sugars can be. If the type of sugar is A, means that is in turn is regulated by an allele. That allele we are representing here IA. IA, which codes for a particular type of sugar which is coming under the category of A type of sugar. So if on the RBC, if the sugar is of type A, means that definitely will be regulated by an allele IA. I stands for isoagglutinogen. It is an antigen only, isoagglutinogen. Then if on the RBC, if the type is B, means then we will that will be regulated by isoagglutinogen allele B. And there are some RBCs which can be devoid of either A or B, then that type of that is recessive character. It is unmodified allele only, so that allele can be represented by IO. So in human beings, instead of 2 alleles, there are 3 alleles instead of 2 alleles. Normally, Mendel he said each and every character is controlled by 2 factors, but this is a deviation only. That is, instead of 2 factors, there can be 3 factors, or now we will call it as 3 alleles: IA, IB, and IO. And when we look into the relationship or the dominant recessive relationship, when we are saying I is completely recessive to A as well as B type of sugar. So on the RBC, as a result, since they are recessive, they cannot produce any antigen, or that antigen will not be, or the sugar molecule will not be there on the RBC. It is completely recessive. It is lacking. It cannot produce any sugar. And if we are considering the relationship between A and B, we have to say when A and B allele is coming together, they express a kind of relationship that is codominance. That means none of this allele is not ready to be suppressed. Instead, both will simultaneously express. So as a result, in AB blood group patients, persons, in their RBC, 50% of the RBC they will have isoagglutinogen A type, and the remaining 50% of the RBC they will have isoagglutinogen B type. So hence that particular type of blood group is called as AB blood group, and their genotype will be IA, IB. So related with this, many problems they can ask. So this example we can give it as codominance, example for codominance. So AB blood group alone will come under this. But if it is A blood group, that means IA, IO, if it is coming, means A is completely dominant over the expression of the IO. So that is why that person can be called as IA blood group. The same way IB, IO, when they are coming, there is a dominant recessive relationship. So B blood group we will call it. And in that way here, when A and B together means AB blood group, that is a typical example for codominance. The same way, codominance, another example only, sickle cell, carrier of sickle cell, HbA, HbS. That is, carrier means 50% of the RBC will be normal type, and the remaining 50% of the RBC will be of recessive type. So HbA, HbS will be acting as the carriers for the sickle cell trait. So that also is an example for codominance. Now we will move on to the basic idea behind the dominance. So Mendel he said always there will be 2 factors. One factor, if both the factors are there, means they appear for that particular trait. It can be a dominant factor or it can be a recessive factor. If it is dominant factor together appears, means that particular trait will be expressed in the form of, in the the purest form. And if it is recessive means that also will be expressed in the recessive expression. Now we'll move into the theoretical aspect. So here in P plant, that is different conditions only we are giving, that is incomplete dominance or this one. Here the first one we can say it as the P plant. So in P plant, the capital R and capital R represents the purple color. And if it is small r means the flower will remain in the form of white color itself. So here in dominance, complete dominance, that is in P plant, directly this white color, the purple color is due to a particular pigment, purple pigment, anthocyanin pigment. So this anthocyanin pigment is produced by a pair of normal alleles, that is capital R and capital R. So if both the, in P plant, if the allelic combination is small r means that white pigment cannot convert into purple color because both the alleles are recessive. Both the alleles are recessive. Hence, the white color will remain as it is. If both the alleles are recessive means the flower will remain as recessive itself because the anthocyanin pigment will not be produced because and the flower will remain as white itself. But if at least one R, capital R is dominant means they can directly, because they can produce anthocyanin, hence the white color completely will turn into pink color. And if both the alleles are of normal R also, in P plant it can directly convert into normal color. That is why there is no intermediate color in P. But in incomplete dominance, when we are coming into incomplete dominance means white and red. Here I have given pink only, that is just for identifying. So if it is a normal allele, normal expression means white, even one dominant allele is enough to convert the white pigment into purple pigment. That is why in P plant there is only two contrasting forms, either white or pink. And that pink anthocyanin formation require only one dominant allele. So even if the other one also recessive also, they will express in the form of purple alone. But when we move into incomplete dominance, there a slight deviation, that is white will convert into pink if at least one allele is normal, and then this pink can convert into incomplete dominance means red flower, into red if the other allele also is normal. So if capital R means the flower will remain as red. And if these two normal alleles are absent, that means small r, this case, if it is small r means that pigment formation cannot take place. So that is why here small r cannot produce the pigment, so it will remain as white itself. And even here also if it is small r means again it will remain as white itself because they are not able to, they cannot produce the enzyme, right amount of or the correct amount of enzyme they cannot produce because both the alleles are recessive. So that is why small r combination remain as white. And the intermediate we have seen in capital R smaller, that is in heterozygous. So why it is so means here a blended expression. So here one allele is normal and the other allele is recessive. So the white color in the presence of a normal allele, that is 50% of the allele is normal. So in the presence of that they can produce, that they are converting into, when we are mixing the red and white, you will get a pink color. The same way here only the pigments will start producing the color, and that is with the help of 50% of that is the allele, capital normal allele can act on it. As a result there can be production of the colored pigment, but that color need more amount of the enzyme, So if there is deficiency of that enzyme which can convert the white into pink, red, if that enzyme amount is less means sometimes it may remain as pink itself because it was not getting the right amount of enzyme. Only the half amount of enzyme or 50% of the enzyme only were present. So this pink, white color transformed into pink, but it cannot completely expressed in the form of purple or red, we can say, if it is snapdragon. So it is remained as the white itself. So one of the main reason is if both the alleles are dominant, the correct amount or the normal amount of enzyme will produce, the white will directly convert into pink or violet. In pea flower means convert into white. Even if it is small r also, in pea plant, that 50% of that one, that means once the enzyme, the presence of enzyme itself can convert that one into purple. So the other one is small r also, because already that much amount is enough for converting white into purple. There won't be any intermediate color. Okay, there won't be any intermediate, whether yes or no. If it is yes, means white will convert into purple. If it is no, means white will remain as white itself. So such type is complete dominance. It is called as complete dominance. So in incomplete dominance, we have to say one allele, they will represent less particular amount of enzyme. The other allele, if it is that also dominant, means the full amount of or right amount of enzyme will be produced. That is why there will be a gradual transformation, that is from white to pink and then to red color. Okay, red or violet color will be produced. So if it is both alleles are dominant, then you will get the dominant trait. If one is recessive, then you will get an intermediate trait. And if both of them are recessive, means it will not convert, it cannot produce color pigment because enzyme is absent. So the dominance is mainly depending on the presence of normal allele, or sometimes as a result the normal enzyme will be produced, or if the allele is less, one allele is recessive, means they can produce only less or non-functional enzyme. If both the alleles are recessive, modified allele, we can say, if both the alleles are modified allele, means they cannot produce enzyme at all. So this is the fundamental basis behind the concept of dominance. Okay, completely dominant means the white completely it can turn into purple or red, that is normal expression, normal amount of enzyme. So white completely convert into red or purple color. And if it is less amount or half the amount of enzyme only is producing, because it have one allele is normal and the other allele is modified. So half the amount of this one only, or non-functional enzyme, they will remain in an intermediate position, that is white color. Not white, pink color. The white will convert into pink, but that pink to red, that much amount is lacking. Amount of enzyme is lacking, so they remain as pink itself. And both alleles are modified from the normal expression, that is recessive, means they cannot produce enzyme at all, and the white will remain as white. I hope with this the fundamental basis of dominance is clear to you. And the same way only in almost all the inheritance pattern, how the alleles are regulating the expression of a particular trait. Now we will move on to the third concept, that is multiple allelism. This we have learned, a part we have learned in codominance. Here, that is mental, he said each and every character is controlled by a pair of factors. But here again a deviation. There are many situations, life situations or in examples around us. We can see in a population, always multiple allelism can be observed in a population. In an individual, we cannot observe multiple allelism, because there is only each and every organism will get a pair of factors or pair of alleles. So 3 alleles it cannot exhibit. So this multiple allelic inheritance, instead of studying in a person, we can study only in a population, because it will be more than 2 allelic forms can be observed, not in single individual, but in population. So here the definition is, it is a phenomenon in which a single character, you have to specify, a single character is controlled or governed by more than two alleles. So it can be three alleles or four alleles depending upon. And here we have to learn only ABO blood group. In ABO blood group, the blood group is mainly determined on the presence of a special type of sugar, isoagglutinogen we will say, on the plasma membrane of RBC. So if on the RBC, if it is of type A, means I stands for isoagglutinogen. So if the type is A, means that allele will call it as, represent it as IA. If it is B type, means IB. And if it is no sugar on this isoagglutinogen is absent on the RBC, means that allele is IO. So here is the pictorial representation of blood groups. So based on this three distribution or three pattern of inheritance of these three different alleles, we can categorize the entire population into four categories. That is A blood group, B blood group, AB blood group, and O blood group. In A blood group, if you are saying, means they can appear in two genotypic ratio. The phenotype is A blood group. The genotype can be of two types. That is, either they are pure for that particular antigen. That is, in all RBCs will have the A type antigen. That means they inherit the allele IA. If IA genotype, means definitely in their RBC, all the RBC will consist of A type of antigen. But there is another possibility is there. That is, since a person is getting the alleles or the genes from their parents. So if from one parent, if he is getting IA allele, and the maternal parent or the other parent, if they are getting IO allele. So when IA allele and IO allele is coming together, A is dominant and IO is recessive. So the physical expression will be 50% of their blood RBCs will have isoagglutinogen A type, and the remaining 50% will not have any antigen at all. But even then, their group, when we are categorizing, they will be coming under the group of A blood group. So here, two genotypic expression for A blood group type of phenotype. The same way, B blood group. B blood group also, if they inherit both dominant IB type, means they will be called as B group, as well as if they at least one allelic representation of IB and IO, means they also will be categorized under the phenotype of blood group B. Then third one only, that is AB blood group. Here only they shows there is no recessive dominant relationship, because IA allele is coming next to the IB allele. So in their blood RBCs, all the RBCs will have IA antigen as well as IB antigen. That is, these two antigens will come on the same RBC. So these both the type of antigens will be present. So they will be categorized as IA and IB blood group. Then the last category is O blood group. O blood group means they don't have any, both the alleles will be recessive. That means IO, IO. So they cannot express any antigens on the RBC. Hence they are categorized as O blood group. So here when we are representing, there are 4 phenotypic expression and 8 genotypic expression. So phenotypic expression means in blood group, A blood group, B blood group, AB blood group, and O blood group. And genotypic expression we are saying means A blood group, IA, IA or IA, IO, B blood group, IB, IB or IB, IO. Then AB blood group, only one expression, one genotype, IA and IB, they are codominant. Then O means completely recessive. O blood group is completely recessive, only IO, IO. So 4 phenotypic expression, but 8 genotypic combination they can express. So with this, deviations of Mendelian we have completed. Then we will move on to the dihybrid cross. So It is just a revision only. So dihybrid cross means it is a cross between two individuals of a species in which two contrasting pairs of characters are studied. And here typical example is it is the cross between round yellow pea seeds with wrinkled green seed. So round is the seed shape, yellow is the seed color. So round and yellow both dominant when they are crossed with double recessive parent, that is wrinkled and green. Here is the pictorial representation. So when if any dihybrid question is based on that is asked, means you have to write the typical representation. That means on the left side P1 generation, G1 generation, F1 generation, then F2 generation with phenotypic ratio and phenotype as well as that ratio you have to write. So here we are representing round yellow, capital R, capital R, capital Y, capital Y. Wrinkled green, small r, small r, small y, small y. Their gamete encircle, capital R, capital Y. From each one a representative. Here small r, small y. Then when we are crossing F1, you have to use the scale and you have to draw the line in this way. And when you are getting it, you will get a round, which one? Round yellow. All F1 will show round yellow. And by selfing F1 into F1, on the left side, you have to write the female gametes in circle. That is very, very important. And on the top, you have to write the male gametic combination. When you are plotting this Punnett square, you will get, you see, a pattern is there. That is from here till this all round yellow. Then again, by leaving alternate columns, another triangle you will get, that is round green. That is this one, round green. Then the next one it is, here it is round wrinkled yellow, and this one is wrinkled green. And these three, that is this one, this three, and this one, this one, this one. So here it is one triangle, complete triangle you have to draw. And this one is alternated. By leaving this one and this one, you will get another small, it's not triangle, but even then like this way you will get. Then the innermost triangle is round green, wrinkled green, you can say round green. Here it is round green, and here it is round yellow. This one is round yellow, and here it is wrinkled green. Okay, wrinkled green. Here it is round green, and here it is wrinkled yellow, and the whole thing is round yellow. Okay, whole thing, that one full complete continuation is round yellow. And this one is wrinkled yellow, green, wrinkled yellow, wrinkled yellow. This one is round green, and this one is wrinkled green. Like that way you can, if you are, but you have to follow the same, capital R, capital Y. Here it is small r, small, capital Y, then capital R, small y, small r, small y. That order if you are writing, then only you can construct this triangular form. Okay, now last you have to write F2 generation. First phenotype, round yellow, round green, wrinkled yellow, wrinkled green. That is 9:3:3:1. So this is also very important. Different combinations of the characters. For that you have to be very, very familiar with which character, which trait is dominant and which trait is recessive. That combination always they can ask. The same way test cross also they have conducted. That is, if it is the same one, test cross means round yellow, F1 round yellow with wrinkled green. In that, instead of 1:1 ratio, since two traits are true characters are participating in that cross, the ratio will be 1:1:1:1, and all these combinations in 1:1:1:1. That I am not representing because we have already dealt with the monohybrid test cross ratio. So dihybrid test cross ratio, no, not much difference, because here you will get this type of gametic, and the other side you will get only small r and small y. So only one row only will be there. Instead of this, you need not replicate again, only this one with crossing this one. So you will get all different combinations, and 1:1:1:1. Now we'll move on to the next one, pleiotropy. This is also another type of deviation only. Here the definition is very important. one single gene can exhibit multiple phenotypic expression. So one gene can exhibit more than one expression. Then that type it is called as pleiotropy. Normally one gene exhibit one phenotypic expression only. That means capital R means violet alone. But here one gene itself can exhibit multiple phenotypic expression. That type it is called as one gene many expression. Okay, one gene many characters can be controlled. So it is pleiotropic. Typical example is phenylketonuria. So what do you mean by phenylketonuria? It is an inborn metabolic error. What do you mean by inborn? That means right from the birth, if that gene is defective, then that condition is called as type of disorders are called as inborn metabolic error, because it is metabolic only. A chemical conversion will not take place, and that is right from the birth because of the deficiency of that particular gene. Okay, so here normally in a normal person, as a result of many metabolic process, phenylalanine will be formed. It is an amino acid only, phenylalanine. And phenylalanine in the presence of normal gene, suppose here if you are writing capital A, if that gene is normal means the phenylalanine will completely convert into tyrosine. And tyrosine is another amino acid which is required for the formation of thyroid hormone and as well as for the formation of melanin, and like that like that many triiodothyronine and all last year we have learned. So thyroid hormone, melanin, etc. So many intermediate components will be formed from tyrosine. So, and this mediation, this chemical change occurs in the presence of a special type of enzyme. That enzyme is called as phenylalanine hydroxylase. So this phenylalanine hydroxylase in turn is controlled by a dominant allele. That only I said, either you can say P, capital letter P, or you can say capital letter A, any letter you can use for the dominant nature of this. So here if it is capital letter P, here we are using phenyl, we can use P itself. Here if it is capital letter P means here if capital P is there means the right amount of enzyme will be produced, and as a result phenylalanine will be completely converted into tyrosine. That accordingly either they will be utilized for the production of thyroid hormone, or they will be utilized for the production of the melanin, which for the skin coloration. Thyroid hormone or metabolic rate will be controlled, and melanin is required for the proper skin coloration. Okay, so accordingly immediately they will be converted either in any of these forms. But if a person is not inheriting he is inheriting a defective gene, that means small p. If he is inheriting small p means defective gene from his parents, then in his body the phenylalanine hydroxylase will not be hydroxylase, okay, XY only, hydroxylase will not be produced. So as a result what will happen, since the absence of enzyme, the phenylalanine cannot convert into tyrosine. Instead of that they will go for another route. That means they will be converted into phenyl keto acids. Phenyl pyruvate, generally we will say phenyl pyruvate, or we can generally categorize them as phenyl keto acids. So this keto acids cannot be converted into any, it will be accumulate in our body, especially in the liver, and from the liver to the kidney, and then it will be eliminated along with the urine. So when it is eliminated along with the urine, within the urine it will contain more amount of phenyl keto acids, and that will give a foul odor to the urine and a smell, and at the same time they express in different forms, that is hypopigmentation of the skin. That means on the skin, because from the tyrosine only the melanin should form. So in the absence of melanin in our body, here and there the melanin deposition will be deficient. So here and there patches of light coloration will appear. Even the same way hair color also melanin, that is why your hair is black in color. So because of that melanin, deficiency of melanin, the hair will become somewhat brownish in tinge. Then this phenyl keto acids, they will be accumulating in the brain. and that is mainly in the brain, in the sense means cerebrospinal fluid. So that can cause mental retardation. So all these are the different expressions. Okay, so the absence of phenyl alanine hydroxylase enzyme, or the deficiency of phenylalanine hydroxylase enzyme due to defect in that trait, in that gene. So if the gene is defective, enzyme phenylalanine hydroxylase cannot be produced in right amount. As a result, phenylalanine will not be converted into tyrosine. This will accumulate in the liver, and then it will be eliminated. It cannot be absorbed by the kidney and is eliminated through urine, which causes elimination through urine and mental retardation due to accumulation of these keto acids in the cerebrospinal fluid, and at the same time it will be expressed. So many characters: hypopigmentation of skin and hypopigmentation of hair, decolorization of hair, and keto acids deposition in the cerebrospinal fluid, as a result mental retardation, and then elimination of excess amount of phenyl keto acids through the urine. So nearly 4 different expressions it can exhibit because of the deficiency of, or the mutation in that particular gene. So this is called as phenylketonuria. Nuria means elimination through urine. Phenyl keto acids are eliminated along with the excess amount is eliminated along with it. So they will have the urine, this patients, their urine will produce a mousy odor. Mousy means elida manam. So that from that, that is one of the main symptoms of that one, the always urinating places and all, that mousy smell will retain. So that indicates he is having phenylketonuria, and it is also called as a type of inborn error of metabolism, because this gene is defective right from the birth. As a result, metabolic process will be interfered. Now we'll move on to another category, that is polygenic inheritance. What do you mean by polygenic inheritance? That is, a particular character can be controlled by more than 2 genes, more than one gene. Okay, more than one gene, just reverse of pleiotropic. The traits are generally controlled by 3 or more genes, are called as polygenic traits, just opposite of pleiotropy. Pleiotropy, one gene regulates many character. Here it is, many gene regulate one character. Okay, many gene regulate one character. So polygenic inheritance. It is also called as multiple factor inheritance. What do you mean by multiple factor inheritance? That is, here each one we will explain it with the help of example. That is skin coloration in the human beings. And here, second point we have to say here, to a certain extent, influence of environment also will take part. The skin color, it is controlled by, in human beings, controlled by 3 pairs of genes, that is capital A, capital A, capital B, capital B, capital C, capital C. If they have that 6 alleles of dominant, means that person will be completely dark. And if all the 6 alleles are deficient, recessive, means they will be very fair. So complete dark, that means each allele will represent or will convert or will produce a particular amount of melanin. So if all the 6 alleles are dominant, means the whole amount of melanin will be deposited and the black pigment will be deposited in the body, and as a result the person will be completely dark, negroid combination. And if all the alleles are recessive, means that melanin production will be less, absence of melanin. So the skin color, tone of the skin color will remain very fair, white, and the pure white will be produced. So here we can see each allele will contribute a particular amount to the total expression. Then hence it is called as additive effect. Okay, that effect is called as additive effect. So that only occurrence and are spread across a gradient, and that effect of that allele is called as additive. So here this is the graphical picture. A complete black person means all the 6 alleles will be dominant, and when they form the F1, that is not F1, here it is G1, there is a reversal only, G1, when I represented by mistake, it is gone. Here it is G1. When they produce the G1, one representative will go into the gamete, that is capital A, capital B, capital C in circle. And on the other side, you see, all it is recessive. So 1 will go small a, b, c in another circle. And when they cross together in F1, here it is F1 only, F1 you will get capital A, small a, capital B, small b, capital C, small c. That means all mulattoes, intermediate color. So what does that intermediate, why it is intermediate? Because out of 6 alleles, only 3 alleles only dominant. So 50% of the melanin will be produced. The remaining 50% they are recessive. The remaining 60% they are recessive. Hence they are not very dark like the black parent, or not. They are not very fair like the white parent. So they will have the F1 generation, all they will be of intermediate character. That is called as mulattoes, or intermediate skin color, we can say. And one mulatto with another mulatto if we are crossing. That is capital A, small a, capital B, small b, capital C, small c into capital A, small a, capital B, capital small b, capital C, capital C. When they are crossed together, you will get 8 different gametic combinations. How we will get 8 different? Because 3 factors, 3 genes are involved. So, and it is occurring in a person in 2. So the ratio is 2 to the power of n. So here 3 alleles are participating. So 2 to the power of 3 means 8 gametic combination can be given, will be resulted. And all these gametic combination on the male parent and female parent, when you are plotting, you will get the ratio 1:6:15:20:15:15:6:1. That means 20 will be mulattoes. Then this extreme left side, the 1 will be exactly like this combination. And on the right side, exactly like this combination. So you will get a gradient. You see, gradient. 20 of intermediate. Both the extremes, one is very fair, the other one is very dark. Then these two, 1:6:20:20. Here it is 1:6:20:20. 20:15:6:1. Here just for representation, only 3 only they have given. So you will get a bell-shaped curve. A gradient will be you will get, and a bell-shaped curve you will get. So this type of inheritance pattern is called as polygenic inheritance pattern. Polygenic inheritance pattern. Why it is called as polygene? One character, that is skin color, is controlled by more than one pair of genes. One skin color is character. It is controlled by more than one pair of genes, that is A, B and C. And the gametic combination is 2 to the power of 3, that is 8 gametic combination. And it is why it is called as multiple factor inheritance, because than normally 2 factors only, here it is 6 factors are controlling. So multiple factors. And it is additive effect also, we'll say, because this one, all the factors are dominant. Then in this 6, 1/8, 1, 2, 3, 4, 5, 6, 7, 8, 1/8 of the melanin will be less. And here 2/8 of the melanin will be less. Here it is 3/8 of the melanin will be less. Like that way, the amount of little by little, the melanin will be reduced. And when we go to that extreme right side, the last case, that is absence of melanin at all. So here the melanin will not be present. So that is why it is given in this way. If it is, when it is moving, melanin pigmentation dominance is going on, it will be increasing, and this side, when we are going to this side, complete melanin, then gradually the melanin pigmentation will decrease, and the last one purely white. So with this we can conclude today's revision session, and next class we will study about linkage and linkage and other important factors. Now we will move on to the next one.